Hi friend, welcome to PLANTI.com. Today I am going to demonstrate the structure of protein in biochemistry. Protein structure. What is protein structure? Proteins are unbranched polymer constructed from 22 standard alpha amino acid. 22 standard. I previously said that 22 standard amino acid present in mammalian cell and more than 500 amino acid present in non-standard amino acid. They have 4 levels of structural organization. One is primary structure, secondary structure, tertiary structure and quaternary structure. Primary structure. The primary structure, the amino acid sequence is specified by genetic information. Amino acid sequence. This is one amino acid. This sequence made the primary structure. The amino acids are connected by peptide bond. Freely amino acid can stay, and when it bond peptide with peptide bond, connected with peptide bond, this form primary sequence. Primary structure. Primary structure of polypeptide determines the higher level of structural organization. Higher level of means secondary, tertiary and quaternary whole structure are determined by this type of primary structure. The primary structure understanding is very important because many genetic disease results in protein with abnormal amino acid sequence. If abnormally amino acids stay, it cause disease and the improper folding and loss the normal function. Okay. Secondary structure. Secondary structure refers to local folded structure that form within the polypeptide due to interaction between the atom of the backbone. Atom of the backbone. Backbone, it is interaction between backbone. Here amino acid sequence, it is primary structure. When primary structure backbone is interact, this form secondary structure. Types of secondary structure: helix, seed, turn. Mainly alpha helix, beta seed, beta turn. It can be gamma helix, beta helix, alpha helix, pi helix, seed, beta seed, and turn also be delta turn, gamma turn, beta turn, alpha turn, and pi turn. Helix. Helix is a secondary structure. Helix is rigid rod-like structure that forms when a polypeptide chain twists into a helical conformation. In helical conformation, it twists in helical conformation. This helix. It can be gamma helix, beta helix, alpha helix, and pi helix. This helical conformation was proposed in 1950 by Linnaeus Pauling and Robert Corey. Formulas of helix. Here I simply draw. Here it is a helix. Here amino acid present. Here in difference between two amino acid. Here two amino acids, suppose this one, these two amino acid difference is called rise. When two amino acid difference are called rise. And a turn, this turn length is called pitch value, and whole the structure length axis, this is axial length, and this is diameter. here you see that this is diameter, this x is that's axial length, and this is pitch value, one turn here, and this is a pitch value, and two amino acid. Here amino acid are frequently here made this secondary structure, and these two amino acid difference are called rise. Here the formulas here, suppose the question here, one amino acids pH value 6 angstrom, pH value 6 angstrom, and the rise value 2 angstrom. So number of amino acid, what is the number of amino acid? So here you think a turn, suppose a turn, here one turn is called pitch value, and here one amino acid between two amino acid difference is called rise. So if I divided pitch by rise, then I can say the number of amino acid. So amino acid is pitch value by rise, and here pitch value 6 and rise 2. Amino acid, so amino acid is 3 amino acid present here. Helix former amino acids are glutamine, glutamic acid, glutamine, leucine, arginine, and alanine. And helix deformer, this type of amino acid can form helix, mainly this type of amino acid, except proline and glycine, because glycine can't play a role because any side chain, it have no side chain due to which rotation around the bond. Proline does not contain NH form on its side chain. That's why these two amino acids can't play a role, but except two are 20 amino acids can play a role form helical structure. Mainly play role glutamine, arginine, glutamic acid, glutamine, arginine, alanine, and leucine. Then I want to say, I previously said that 4 types of amino acids are helix are present: gamma, beta, alpha, and pi. What is the difference between these 4? Or what is the difference between these 4 types? Helix, when a carbonyl group of one amino acid, carbonyl group connected in after 2 amino acid in amino group, that is gamma helix. When carbonyl group attached in after 3 amino acid, that is not amino acid, amino group, that is beta helix. Here I want to clear that. Suppose one amino acid, here 1, 2, 3, 4, 5, 6. Suppose one amino acid connected with after 1, 2, this connected between it, this is called gamma helix. Beta helix when it joined after 3 amino acid can contain one amino group, and when a carboxyl group bond with one amino group, that beta helix, 3 number amino group, that is beta helix. When 4 number 1, 2, 3, 4, that bond alpha helix. When 5 number, then it bond pi helix. Okay. Here I want to say that obviously 3.6 is alpha helix. It another name 3.6.13 and beta helix name is this name is 3.613 and beta helix this is 310 helix. Gamma helix 2.27 and pi helix 4.416. The most helix is alpha helix. Alpha helix 3.613. Okay. A compact slide. This is type and this is number of residue per turn and this is pitch value and this is rise. Here at first alpha helix, the number of residues per turn is 3.6, pitch value 5.4 and rise 1.5. Here beta number of residues per turn 3 and pitch value 5.8 Armstrong and rise 2.0 Armstrong and gamma helix 2.27 this is 6 Armstrong, pitch value 6 Armstrong and the rise is 2.72 Armstrong and the number of residues per turn in pi helix this is 4.4 and this is pitch value 5.2 and the radius and the rise of the pi helix is 1.1 Armstrong. Okay, this is the all in compact slide. When two or more polypeptide chains segments line up side by side, each individual segment is referred to as beta strand. Suppose this one is beta strand and other one also beta strand. Two segments line up side by side. Two are line up side by side. This form a beta sheet. This beta strand is fully extended. That means alpha helix is helically present, but beta sheet is fully extended, elongate. Okay. The distance between adjacent amino acid means distance between two amino acid at near amino acid. Two amino acid, adjacent amino acid along beta strand is approximately 3.5 Armstrong. This is also you can say rise value. In condition of alpha helix, I say that in difference between two amino acid distance is 1.5 Armstrong. This rise value is 1.5 Armstrong, but here 3.5 Armstrong. Beta sheet are stabilized by interchange hydrogen bonds that form between the polypeptide backbone NH and carbonyl group adjacent strand. It also same as like helix structure because here also one amino acid can bond amino acid in NH group can bond with carbonyl group after 1 or 2 or 3 or 4 or 5 amino acid it can bond. Okay. H bond formation in beta sheet. Here one amino acid C, R group here, one amino group, one carboxyl group. Here also one alpha carbon, here R group, here carbonyl group, and here amino group, here amino group, here carbonyl group. Here you see that one carbonyl group attached with one amino group in this way. And you can see also here amino group present and here carboxyl group present, here carboxyl group present, here amino group present. These two are NC and CN. NC. Two are antiparallelly stay here. N to C and here N to C. In this way, in opposite direction, that's why antiparallel, and it is the more stable structure than parallel beta sheet. I also deeply describe here parallel beta sheet. In parallel beta sheet structure, the polypeptide chains are arranged in same direction. Here C terminal, here N terminal, and here C, here N. And when here C and here N, and here A, here C and there N, that is antiparallelly. In same side, when two C terminal are present, that is parallel beta sheet, and when one C terminal and one N terminal present, this is antiparallel beta sheet. And here you see the bond formation here. Here NH group bond with carboxyl group in this way. This is the loosely bond, and if you see the antiparallel beta sheet, here antiparallel beta sheet chains run in opposite direction, C terminal, N terminal, N terminal, C terminal. Antiparallel beta sheet are more stable than parallel beta sheet because they are fully bonded. Because fully collinear hydrogen bonds form. This hydrogen bond is more stable. Okay, this antiparallel beta sheet is mainly occur. Then come to the point turn. What is the turn? Most proteins have compact globular shape requiring reversal in the direction of their polypeptide chains. Many of these reversals are accomplished by common structural element called the turn. Okay, the turns composed of 3 to 5 residues, glycine and proline are commonly present here. In all other secondary structure cases, their glycine and proline are deformer, but here former, turn formed by glycine and proline. The lack of large site present in glycine, not a large side chain present, because one H present in its large R group, and cyclic structure of protein ideally suits for the turns, and proline have the cyclic structure. They are known as well as reverse turns, hairpin bends or omega loops. The other name of hairpin bends, omega loop and reverse turns also. So what is turns? Here you clearly understand hairpin loops. Hairpin loops also one type of turn. This loop turn, it made by glycine and proline. Loops which connected to adjacent antiparallel beta strand are called hairpin loop. Two residues long hairpin loop are often called reverse turns. Beta turns so simply turns. That means loops connected to this is beta seed, this is beta seed. Two beta seed are present and one turn in present between them. And this like as a loop. This is hairpin loops. Loops which connect adjacent antiparallel beta strand are called hairpin loop, as like hairpin. Two residues long hairpin loop. This is two residue here present. Two amino residue. That means one residue means one peptide bond with one peptide bond. That means four amino acid present here. And it also, if four amino acid present there, it can form two residue of amino acid. Simply one amino acid also can present, and one residue means when it bonded by peptide bond, then it called residue. Two residue present, that means four amino acid present, and it can form turn hairpin loop. Loops have hydrophilic structure, hydrophilic residues, and they found on the surface of the protein. The common word that hydrophilic are always present in the outer side and hydrophobic is present in inner side. Here say that hydrophilic residue and they are found on the surface side of protein. Okay. Here then loops are made up of more than 10 amino acid. Loops are made up more than, in previous case I say that here only 4 amino acid can form a hairpin loops. Then it is also called reverse turns. Okay, but normally naturally 10 amino acid present in a made up of loop more than 10 amino acid. Loops that have only 4 or 5 amino acid residues are called turns. Loops, when a loops only made up of 4 amino acid, that also called turns. They have internal hydrogen bonds and they are by internal hydrogen bond. Reverse turns are a form of tight turn where a polypeptide chain makes a 180-degree change in direction. Here one type of direction and change it 180-degree direction means it opposite to other side, and this is 180-degree direction, and this is called reverse turns. When one turn here, 2 amino acid suppose present and it turn 180-degree. Here 2 amino acid present. They are internally hydrogen connected by internally hydrogen bond. This is reverse turns. Reverse turns also called beta turns because they usually connect in beta seed. Why it called beta turn? Why it called beta turn? Because two beta seed, here one beta seed. You can call also beta strand. So these are connected by beta turn. Two beta strand when connected by a turn that call beta turns. Two beta seed when connected by a turn that call beta turn. Okay. Here types of turns: delta turn, gamma turn, beta turn, alpha turn and pi turn. Here is also same case. One amino acid when bond with one amino acid, 2, 3, 4, 5. If bond, this NH bond, suppose bound here carboxyl bond, CO bond. Here this is called delta. NH bond with one after one amino acid. Here one amino acid bond with next amino acid. That is delta turn. When bond is after two amino acid, 1, 2. After two amino acid, when it is bond, this is gamma turn. This is delta turn, this is gamma turn. When bond is after 3, 1, 2, 3 amino acid, this is beta turn. And when bond is after 4th amino acid, this is alpha turn. And when bond with 5 amino acid, this is pi turn. Okay. In this way, they are connected. This is the type of turns. And it also remember that glycine and proline are the transformer are glycine and proline, and other except 2 amino acid, other all 20 amino acids are transdeformer. Then come to super secondary structure. Here a specific geometric arrangement of alpha helix and beta sheet. Two are alpha helix and beta sheet connected through loops are called super secondary structure. It can be loops, turns. Okay. Super secondary structure is not the part of secondary structure. It the third part of amino acid structure. In between secondary structure and tertiary structure, its mediatory structure is super secondary structure. That is when two, suppose alpha helix, here only helix present, and when beta sheet, only beta sheet present. But when only turn, so only turn present. When two are present in a same case, that is super secondary structure. That means, suppose it also called motifs. It comprises at least 2 units of secondary structure, at least 2. Suppose one helix, one turn and one helix. Here 2 are present, one helix present and one turn present. 2 types. Helix is one type and turn is 2 type. Suppose in other case a beta sheet present and one loop present. Here also a beta sheet present. So here also 2. One beta sheet present, one type of beta sheet, and one is loop present. This is 2. This is the super secondary structure. It can be 3 or 4. Here also can be alpha helix, helical structure, alpha helical structure. It can made by 3 form: one beta sheet, one loop, and one alpha helical structure. Here some example. Here helix, turn, helix, helix, turn, helix. Here helix, alpha or beta, gamma, कुछ भी. Helix, this is turn and this is helix. Here helix, loop, helix. Here you see that one loop present. Helix, loop, helix. Here coiled, coil. Here coiled form. Coiled domains also bind DNA. Individual helix have heptade repeats. Here coil are present. Beta hairpin. Here beta present and here this loop is like hairpin. Okay. Here also Greek key, beta sheet present, and this is the turn are present, and it make like Greek key, that the name Greek key. Beta barrel, it can form beta barrel, beta sheet, beta turn, can bind beta barrel. Beta alpha beta sheet present here and alpha also present here, alpha helix, that can form beta plus alpha motifs. Okay. Then next, tertiary structure. Tertiary structure is the 3-dimensional conformation of polypeptide. three-dimensional. At first I say amino acid present freely, then amino acid can bind by peptide bond, then they are bind by hydrogen bond. After 1, 2 or 3 amino acid it can bind, and it is secondary form. And when tertiary form, it is three-dimensional conformation of polypeptide. The common features of protein tertiary structure reveals much about biological function of the protein and their evolutionary origin. That means one amino acid sequence can't play role in function. They are also important in playing role, but they cannot properly a role play in function. When this type of amino acid sequence meet and make a tertiary structure, this tertiary structure can't play a role in biological function. Which tertiary structure. The function of a protein depends on its tertiary structure. If this is disrupted, it loses its activity. Suppose where you see that protein disrupted, that means is not amino acid disrupted. That doesn't mean amino acid disrupted. That means a protein tertiary structure disrupted, the bond disrupted. This is the tertiary structure, three-dimensional structure. Here tertiary structure, how it form? When secondary structure are fully coiled and folded. How it folded? It is folded in a manner. If it is misfolded, it can cause a disease. Severe disease. But when it is properly folded, it is okay for a play functional role. Precise 3D shape. Folding is determined by interaction between R groups. How it folded? By interacting R group. It can fold it. Okay. How it? Here you see that one hydrogen bond form here. It can form hydrophobic interaction. It can peptide. It is also peptide backbone. Peptide bond also here. And this is disulfide bond. This is very important bond, disulfide bond. And this is ionic bond. It can form hydrogen bond, disulfide bond, ionic bond, and hydrophobic bond. Hydrophobic interaction. This type of bond are present and make a proper folding. This bond playing a role, vital role play this bond. The secondary structure become a tertiary structure by this type of bond. This bond can fold a protein properly. This and make the protein tertiary structure. Domains means fundamental functional unit of three-dimensional structure of protein. Suppose here one functional unit, two functional unit, and three functional unit. Three type of functional unit are present here. Domain 1, domain 2 and domain 3. Here this is also a tertiary structure, but here three types of secondary structure are present and make a domain, three domain make a tertiary structure. Domain often have specific functions such as binding of small molecule. Specific structure that can binding a small molecule. This is the main playing role. It is the functional structure of tertiary protein. Quaternary structure. The biological function of some molecule is determined by multiple polypeptide chain called multimeric protein. A biological function of some molecule that determined by multiple polypeptide chain. Here that means one tertiary structure when two tertiary structure, two or more than two, next time read here, and each polypeptide chain is called subunit. Subunits in a multimeric protein may be identical homomultimer or quite different heteromultimer. Arrangement of polypeptide subunit is called quaternary structure. Subunits are held together by non-covalent interaction. That means two tertiary structure or more than two tertiary structure. Tertiary structure means that also a polypeptide chain. Some molecule determine the multiple polypeptide chain that that can make quaternary structure. It can be homomultimer and heteromultimer. Suppose one alpha unit present, and these two polypeptide chain, this alpha polypeptide chain and this alpha polypeptide chain are homomultimer. This is homomultimer. But when two are different, quite different, this is alpha and this is suppose beta present. This is different, alpha and beta. This is heteromultimer. And each polypeptide chain are called a subunit. This is the quaternary structure. Okay. Thank you for watching my video. Thank you.